on with the next exciting talk in our session. It's by Ben Todhunter from the New Zealand Merino Company Limited and Cleardale Station Limited, and Dr. Mark Ferguson from NeXtgen Agri International and Genesmith, New Zealand, who are going to be talking to us about genetic approaches to reducing foot rot in sheep. So Ben and Mark, I think, are both doing the presentation. If you already shared your screen, so if you'd like to take it away, please.
Well, thanks, Hannah. And it was a fascinating presentation from Lynn, and just with the industry, I think this is an example where we have been able to do that with aligning animal welfare, animal production, and industry goods. So, a very fascinating presentation. So kia ora, Ben Todhunter. I come from the middle of the South Island of New Zealand. We farm around about 6,000 breeding ewes, 3,000 hoggets, and we got foot rot in about 1993, and foot rot's a disease of ruminants. It principally affects sheep and cattle. It starts off between the toes, and then follows up the axial groove, and then leads to underrunning of the foot, and significant pain and welfare issues in the animal. So the end result you'll get with an animal is it won't be able to walk, and will often have to be euthanized, and the production goes down. So there's about 1.2 billion sheep in the world. So if we can improve the welfare of even 1% of those that have foot issues, there's a significant benefit to welfare of animals around the world. As I've said, the disease starts from the hoof and comes out and leads into significant lameness and the other one. So if we go through to the next slide, Ferg, we'll look at that one. So the traditional approaches to dealing with foot rot follow a solution-like approach, and they don't dissolve the issue at all, and they all come with their own complications and issues. So one of them is troughing the animals 3 times for 20 minutes in a zinc sulfate solution. The other options are vaccination, antibiotics, or turning the sheep over, trimming the feet, and isolating the foot rotty stuff. None of these approaches to the disease eliminate the problem at all. So you are constantly dealing with the pain of the animals, the people issues of trying to deal with the sheep, bringing the animals into the yards to handle them, and the production issues that go with the lameness and those handling things over time. So history looked at trying to find a solution or to dissolve this issue. We're very happy to get behind it and support it. So I'll hand over to Dr. Mark Ferguson, who's helped us a lot with this.
Thanks very much, Ben, and thanks all, and thanks to the organising committee for having us on this evening, where I am at least. So yeah, I've moved from Western Australia to New Zealand in 2012 to sort of start this work. And we were supported by the Ministry for Primary Industries and New Zealand Merino Company as the 2 sort of core sponsors, and support of Merino Inc. And so we pulled this together as Unlocking the Perfect Sheep was the title of the project. So fundamental to us, any scientific approach, as we've just heard, is a scoring system. And so there already was a scoring system for feet. And we were utilising that. So 0 being a perfect foot, 1 where you've got a bit of water maceration but no infection. Score 2 where you've got some infection between the toes but only in the skin. Score 3 is where you classically have got a foot rot lesion, where you start to get the hard material, the keratin material, being impacted by the bacteria. Score 4 is where you get to that underrunning actually runs right across to the other wall of the foot. And score 5 where it runs around to the toe and gets towards that one that the slide we showed earlier, with a really basically rotten foot, which is hence the name of the disease. So as part of this scientific effort, we scored literally hundreds of thousands of feet. So I've seen a lot of sheep feet over the last 10, 11 years, and basically we were looking for a solution that breeders like Ben could use, that they could a way that they could use this technology to go forward and select the animals that were better. So essentially we were collecting DNA, we were collecting pedigrees of the animals, so knowing who mum and dad is and grandparents to look. That's how genetic, obviously, evaluation works. Like in any disease challenge, you have to actually, or disease resistance work, you actually have to have the animals under challenge. They actually have to express the disease and to allow the susceptible ones and resistant ones to differentiate. And then so there was under a foot rot challenge, which is not that hard in the South Island of New Zealand in most years. As in it's an endemic disease that is often there. And then we were obviously measuring other performance factors because we wanted an animal that was both resistant to disease but also do all the other things they need to do on a farm. So essentially we're heading towards being able to go to a group of selection candidates and then select the ones that were resistant to the disease and the ones that were, and breed from them and not breed from the ones that were susceptible to disease and move the population. So we knew from work in Scotland and Australia that it should be heritable as a trait. So heritability is the proportion of the variation that's expressed between individuals that's explained by the genes. And so we were really happy when early on in our work, so 2 or 3 years in, when we'd collected enough of the foot data and crunched the numbers, that foot rot was heritable, and it was heritable at a pretty good rate, 20 to 25%. So it meant that we could make genetic gain for that disease. And I guess the language we use in the livestock industry to explain genetic variation is estimated breeding values. And so that expresses the variation of any trait. But so for foot rot, the EBV for foot rot was the critical thing we're heading towards. And beautifully, an EBV takes out the white noise that I call it, which is the environmental variation for the disease, and just leaves us with how much genetic variation is that animal, how much genetic superiority is that animal expressing. So we kind of had hopes originally that we would find a couple of major genes, and so that's awesome if you find major genes being things like eye color in humans or human, not human height, but eye color in human, where both your parents have blue eyes, then you're going to have blue eyes. But early on, a lot of the early work we found that's how much genetic variations on each of the chromosomes across a sheep, and so basically every chromosome pretty much was contributing some of the genetic variation to foot rot. So it meant that we didn't have any major genes to work with. So that meant we needed to go down that estimated breeding value approach. If we had a major gene, we could just do a gene test of the population and find the ones that were carrying it, and away we would go. Unfortunately, that wasn't the case, and was probably obvious, because if it was that easy, then we would have done it. It would have been easier to breed for. So it's what we call a polygenic trait. Lots of genes, hundreds of genes impacting on disease resistance. So if we fast forward to sort of where we are today, we've now got a population that's very well characterised in New Zealand for the disease, or for resistance to the disease. And so if we look at this graph, we've got the -1 there, the very low numbers on the left-hand side of that graph are the resistant ones. So they're the ones that do not express the disease, or are genetically less likely to express the disease. And the ones out on the right are the ones that do express the disease. So and very much so. And so they're very susceptible. And if we look in the population in a breeding value, it starts at 0. So 0 is sort of ground zero, obviously, and the baseline, and then it deviates from that. So I guess by this point we knew that we had a lot of genetic variation, and we knew that there was a lot of and it was heritable. So a great combination that meant we could make some genetic gain on that trait and help lots of animals get into a much better welfare state, which was pretty exciting days, and been a great 10 years really. So what does that mean for the animal? So at a -1, this is, I mean, maybe a bit complicated, but I'll quickly walk through it. -1 is that really resistant animal now, so very low susceptibility to the disease. And so if we generate some progeny from a sire, there'll be sort of 15% will get some bit of underrunning, but none will get into that score 4, and definitely none into score 5. You'll have about half the progeny that will get some score 2, but that's any bacteria can cause score 2. That's just a sort of a disease of the environment almost, when there's wet conditions. Compare that to out here, where we have got animals in the industry still that are +1, and you've got sort of half of the progeny that are got at least score 4, so very, very lame and sore, and almost 100% of the progeny of it got at least score 3. So we've been able to characterise and breeding out towards this direction. And I'll hand back to Ben to sort of talk through how he's used that information.
Yeah, so in 2014, once we understood the science around the disease and we had a tool that we could select for the resistance really quickly, we set up a group breeding scheme with a bunch of farmers, deliberately trying to target the genetics of this trait and improve it, and try and maintain the other productivity traits of the sheep. And the beauty of the foot rot resistance is it's not really negatively correlated to other traits at all. And since that time, within about 3 years, we dropped a sire out that's still one of the top sires in resistance in the database. So we've been very lucky with that. And as you can see, from about 2016 through to 2022, 23, we've made massive progress in the genetics of the database. The graph, it's a graph, but it's a pretty sexy graph for all the people that have done the work around the industry in that respect. And what that means is that we can shift the genetics of our population from, as Mark said with his graph, normally distributed around the mean. We've shifted that distinctly to the left, to the more resistant part of the population. And what that means is it's a bit like compounding interest, is that you have less infectious animals in the population, so you have less bugs in the pasture, so you have less challenge for other animals, and they get less infection. So it's a really virtuous circle. And we've been able to do that in a short time, and move the disease from being something that was really hard on people and animals and production, and had some environmental issues, to a disease that's now manageable for us, and we can take the knowledge and learn to enhance the welfare of other animals in the New Zealand sheep industry and in other industries, sheep industries globally as well. So it's a really good example of where we've had a problem. We've used some really good science to understand the problem clearly. We've worked, farmers and industry have worked together, and now we've got a tool and a system for dissolving the problem, so that we can manage the sheep and have great products that come out of our New Zealand environment and sheep, and we're spending less time bringing them into the yards, inspecting them, and all the other welfare issues that go with that. So yeah, really happy to answer any questions, Anna.
Thank you so much, Ben and Mark. I think that this is a really wonderful example of the science working for the people in the industry. So that's really fantastic. Yeah, someone has a question in the chat already. It's actually similar to the one that I wanted to ask you myself. Do you think that this experiment could be applicable for other hoofed animals, wild animals, or other livestock species?
You asked that, Ferg?
Yep.
Oh, you can.
So yeah, definitely. And so I guess if we look at, even if we stay within species, so the other things we're doing as an industry are working on worm egg count, also reducing worm parasite burdens through the same sort of process, through selection, fly strike, other pretty much. Yeah, I'm pretty passionate about removing diseases through genetics. And so yeah, so definitely in sheep, other animals, so there's examples in cattle, in goats. So probably not, you don't get any selection in wild species, then that's sort of a natural process that happened. They've evolved to get better at handling parasites and handling things in the environment. I guess it's, but in farmed or housed species, then definitely there's always genetic variation for everything. And so it just takes the effort and the investment to get it done.
You've got a few more questions, but I'll just pick one. Random, well, kind of random. It's from Kate, who's also from New Zealand. So what has the interest been from commercial farmers, and have they been prioritizing this trait when they're selecting breeding grounds?
Yeah, commercial farmers who have foot rot, they've been very interested in the trait, and they've actively targeted in selection. And so we've been able to start spreading the genetics throughout the industry quite rapidly for those who have the trait and are interested in it.
And we're now to the point where we're replicating into Australia. So it's gone beyond New Zealand.
That's fantastic. That's so wonderful. Something that I thought about when you were speaking was Ben mentioned, it was causing a lot of harm to the humans as well as the animals, the people who were taking care of them. So I thought that was a really nice inclusive concept of welfare as well. Like, it extends beyond the animal's experience, right? I would love, again, to talk to you a lot more, but I guess in the interest of time, we should thank you very much. And we do have more questions for you in the chat if you would like to engage with them. I'm sure people would really, really appreciate it.